my only interactions with that have convinced me that it's not worth your time. But bioscheme, never heard the term before. Maybe it's applying scheme to problems of life, or maybe it just reflects the fact that for most of you, scheme seems to be your life. I'm going to describe some work I did a while back with Bob Zagurski of the vaccine group at Wyeth Pharmaceuticals. Dave Russell contributed a lot because I don't know how to do databases, but he does. Okay, a little bit about biology to start with, a kind of primer, or primer, whatever we call it. DNA is a double helix. Here's the pictures. It's intricately connected with Dan. Dan and his DNA are, I can assure you, intricately connected. I don't know if you've noticed that also DNA is an anagram of DAN. So it certainly seemed to me an appropriate topic for this meeting. What's it got to do with Dan's 60th birthday? And here I'm going to stretch it, kind of like Bill did yesterday. It's no coincidence that Dan is 60. 50 years ago, the structure of DNA was discovered. Just a coincidence? Might it be that the 10-year-old Dan discovered the structure of DNA? Possibly. Just a coincidence? It didn't happen, but he might have. Okay, some axioms. DNA can replicate, and here's how it happens. The double strand separates, and then each strand can recombine, and on it goes. Some stretches of DNA, actually, I'll translate it into proteins, the stuff of life, and we're going to go into the mechanism for that just a little bit. I don't know if these pictures help any. I'll try and get to some more meaningful pictures as we go on. Here's a little stretch of DNA here, indicating a couple of genes. And some of the features you'll see are codons for producing the string of amino acids. So you're converting from one string, nucleotides, a 4-element alphabet, to strings of amino acids, a 20-element alphabet. We have things like promoters that encourage this to happen. We have stop codons that say stop the translation. And regulation is achieved somehow, and we don't know yet just how. Well, are there any steadfast rules? The laws of physics apply, the laws of chemistry apply, the laws of mathematics do apply too. There's some logic to it. Now, the laws of biology, the only law of biology that I know is that every law of biology has an exception, including this one. That's for you, Doug. Okay, the laws of man, I don't know if they come into it or not, but that was a cool picture anyway. It's my favorite TV show right now. Okay, how do you detect a stop when you're translating DNA? Well, there are 3 possible sequences. Now, when I say there are 3 possible sequences, that is most of the time. Other organisms might be slightly different, but generally speaking, there are 3 stop codons, and whenever the translation process gets to one of these, it stops. And that's pretty easy. Translation process, here's the genetic code, and there's a wonderful article about this. It must be years since you wrote it, Doug. The genetic code arbitrary. Get the initials, TG. It's a very, very clever article. It must be a long time ago. Lovely article to read. Brian Harvey had an article in American Scientist just a month or 2 ago. A great update to it. And it's wonderful, pioneering work that even a first-year undergraduate learning scheme can do some world-shaking research and publishable results. There's really so much low fruit there. We're just waiting to be picked. Use it in your scheme courses. Great stuff. Well, basically what happens is sequences of 3 nucleotides. Now, sometimes one's called U, sometimes it's called T. It's just the difference between RNA and DNA. It's sort of irrelevant chemistry. U and T interchangeable. When you get three of them, they code for one of the amino acids, and the amino acids, I've named them over here. There are standard three-letter abbreviations for them there. And it's very interesting, of course, we've got potentially 64 elements, but only 20 are coded for. And do read Brian Harvey. It's fascinating as to why the code evolved just the way it did. Okay. Of course, the answer is we don't know yet, but the speculations are fascinating. Okay, that's the genetic code. I want to get to the start codons. How do you know to start producing a protein? Turns out there are three usually start codons again. Almost always it's this one, ATG. But notice there's a difference between start codons and stop codons. The stop codons are stop and nothing but stop. But the ATG, for example, A, T, which is now U, because it's RNA in this chart, and then G is methionine. But it's also a start codon. How do you know which it is? Right, there's a bit of ambiguity. Right, we're not talking deterministic finite state automata here. So let's look at the problem of finding offs. There's a rather nice program. I wish it had been written in Scheme, but it's written in Java, called Artemis, that I'd just like to...
Tell people what an orf is.
Oh, I will. When we get this picture up, I should make it fairly clear. Let me bring this up. This is Artemis running right here. You can just ignore that top part for a moment. This is showing part of the genome of E. coli, if you can stomach that. As you'll see, what you see here is sequences of nucleotides, A, C, G, T. This is the forward strand, here's the reverse strand, there's the address within the genome. And here, it's put in the three possible translations of the forward strand. It depends where you start counting threes. So there are three reading frames, as they're called. And these are the one-letter abbreviations for the resulting amino acids, together with the stop codons that can appear here. There are different kinds of stop, too, hence the asterisk, sometimes the sharp sign. Now, it's interesting, as you change the scale of this, let me see if it'll do it, there. There's no room now to put in the actual names of the amino acids. So instead, it shows just the stop codons. And you can see from this description here that there are some stretches with no stop codons within them. And those are potentially coding for long proteins. Okay? They're called open reading frames because there are no stops in there. So one of the first steps in looking for genes, or looking for stretches of DNA that code for proteins and cause proteins to be created, is to look for longish open reading frames. Okay, easy program to write. Find where the stop codons are. Find long stretches in between them. Easy program to write. Let's go back to here again in our keynote and look at a slightly more difficult problem, finding genes. Now for genes, not only do you need stop codons, you need a start codon. And the trouble is, how do you decide which start codon it is? And that is a hard problem. As you can see, I was supposed to run Artemis again. For reasons of time, I'm going to jump ahead, and we'll come back to it if anyone's interested and we have the time at the end. This is a picture of a creature called a ribosome. It's just a big glob of stuff. And the ribosome actually does the translating. And again, Doug's article is one of the best descriptions of how this happens. Basically, you grab three, or you glom onto three little nucleotides over here, and at your end you produce some tRNA. Okay, and that sort of, to each possible triple here, there'll be a piece of tRNA, which it's neat stuff. And again, do read Doug's article. But you can see roughly how it works. There's a piece here that sticks, and then codes for an amino acid up here. It's not quite that simple, but this picture gets across the idea. And the ribosome kind of moves along the piece of DNA like this onto the next triple, and as it does, it creates this string of amino acids, which will make your protein. Okay. How does the ribosome know to grab onto the DNA and start doing its thing? That's the question. Okay, well, we have a few clues. Now I'm going to teach you as much biology as I know. There's a piece called, it's a subunit of the ribosome that looks for a starting sequence like this. It's called the Shine-Dalgarno sequence that says, get started. It's as if a little piece of the ribosome looks for this, gets on there, and when it's stuck, it can start doing its translating thing. And typically, you'll find this characteristic sequence, between about 5 and 30 base pairs of the true start of the gene. Okay, we don't know exactly where. It won't be exactly this. We'll get into it a little bit more. Pearl, you'd think, would be good at finding Shine-Dalgarno regions like that. It's just pattern matching, right? Well, no, it's not. It's approximate pattern matching. And even then, you can come up with the approximate pattern matching tools that you can in Perl, and still it's not right, because then you start looking at, why is it that a piece of ribosome can stick onto something like this? It's probably some kind of physics. Where's Jerry? Okay, there's physics in it. There's electrical forces, all sorts of stuff. There's chemistry and physics going in. Perhaps it's more suitable for Fortran. So these are some of the features that we can look for when we're trying to identify the start sites. We will be looking for something that's a bit like this, because that's the Shine-Dalgarno sequence we're trying to glom onto. Probably we're going to need to look at least a 3-element subset of this in order that it can stick at all. Probably you want more than that. The spacer between the Shine-Dalgarno and the start codon, that is important. The percentage of CG content as opposed to AT, that seems to change when you're close to a coding region. Just how varies from species to species. What kind of size of window do you need in order to determine the average CG content that is relevant to what you're doing? Should you have a big window and average over that, or a small window, focus it in? What's the best way to do it? You're much more likely to have ATG than GTG or TTG as a start codon. How much more? Who knows? Wasted open reading frame is considered undesirable, but why? Okay, and the biologists go for all sorts of reasons. You know, if it's not really coding for anything, then it's probably not very important, and so mutations will survive into the next generation, and so there's a good chance you'll have stop codons at random in there. Who knows? But how undesirable is it? So you can see that it's an ill-defined problem. Okay, going back to Jerry's talk this morning. Can we do the best of it? Well, if we're writing in Perl, no, we can't. It's a mess. If we're doing it in Fortran, we can't. Because those languages constrain the way in which we think. So how do you calculate them? Do you do it in Fortran? How much more suitable is Fortran than Perl? Electrical calculations. There's some work done by Dave Turner and his students at the University of Rochester. They go into the thermodynamics of RNA secondary structure. That's where I found these various pieces of physics that show how there's a context-sensitive kind of way of calculating the attraction between two nucleotides. You can code this much better than my scheme code here in Fortran. You can code some of the searches I do much better than I do in Scheme in Perl. You can code various pieces of my algorithm much better, much more efficiently in other languages. But you wouldn't. That's the whole point. Then we need to know how we're going to combine these various factors. Do they make a positive or a negative contribution? Is it a linear combination? We really don't know enough. And again, if we're in the Fortran mentality, we're just going to try something. We're going to try and find out as much as we can by asking our colleagues. We really don't know enough about what we're trying to do. So let's go to scheme instead. Scheme is equally accomplished for all tasks. I chose that word because it's kind of a euphemism for it's equally good for all tasks or perhaps equally bad for all tasks. But at least you're not driving your work because of your choice of language. Right? The whole Alberson and Sussman, the whole Friedman and Rhys, everything I learned from these great people is that programming language is for expressing ideas, developing ideas, conveying algorithmic ideas. That's what it's all about. It should not get in the way of your freedom of thought. And my experience elsewhere is I'm so worried about where the semicolons are going that I can't think freely. So it's a bit of a fortunate thing about Dan's name. There's also this Fried part. There's a freedom about it, and of course his collaborations with a very wise collaborator. Is it just coincidence? I would say. I think not. Dan did something very valuable for me many years ago, 20 years ago. I was a little bit of a skeptic about scheme. You know, I'd been programming in Fortran for a long time and thought I could do things more efficiently, better. And all Dan did was, as usual, he gave me something to read. It was your thesis, I believe, with Fortran code. And it was the most beautiful Fortran code I've ever seen. And as you read, you think, and you come around. So, what did I do? Well, I figured that if a mildly complex molecular glob, like a ribosome, can solve this problem, okay, with 100% accuracy, the ribosomes get it right every time. And yet the best programs written in C or Java or Perl are lucky if they can achieve 70 to 85% accuracy on the very difficult problem of selecting which is the start site, which start codon is actually the right one. Why can't I write a program to simulate this glob? Now, of course, I don't know enough of the science to do the simulation precisely, but I do know that these globs evolved. So can I write a program to evolve a glob simulator? Well, the glob has had billions of years of evolution to learn its thing. I don't have billions of years, but I do, thanks to Kent. Is Kent here? There he is. Thanks to Kent, I can simulate a whole generation of globs in about 11 hours. It sounds a lot, but we've got weeks and weeks to run these things. I can also speed up the rate of mutation, and with any luck, I can get somewhat closer to a glob than I can to a Perl program. Is that true? Yes, I can. After several months of training, I used the forward strand of E. coli for training. There's a well-established set of verified start sites that I could use for the training. I did all my training on the forward strand, and then on the reverse strand I got to 94%, which is way better than anything we've seen in Perl. Could it have been done in Perl? Of course, you can translate my scheme program into Perl, but would it have been done in Perl? Would it have been done without the kind of free, anarchic thinking that Dan has encouraged us all to do? I think not. So, thanks, Dan. Jim.
There's a guy at Argonne National Labs, Ross Overby, who develops his own
I know
regular expression syntax for matching DNA expression syntax that has operators for, you can say how many mismatches you will allow for a certain subsection. But also the special operator for back references. You know how in Perl you can say, open paren, blah, 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 close paren, and you can say backslash 1 or backslash 2. It's the same thing that I matched back there. He has one that says the same thing that I matched back there, but complemented genetically. That is, it turns each base into its complementary pair. And what this lets you do is it lets you recognize sequences that complement each other and which, when allowed to fold up the way they please, form a helix. So you can actually use this regular expression language to recognize physical properties that the RNA makes when it's in vitro, or when it's in vivo.
Great.
So
Tell me more afterwards.